eukaryotic cells. The first thing it's important to understand is what are these processes achieving for the cell? In other words, what are they doing? Why is the cell choosing to undertake transcription and translation? The answer is that these processes facilitate information coded in DNA being translated into a language that the cell can understand in order to make proteins. Now, this is important because proteins are essential to survival for all eukaryotic cells and therefore eukaryotic organisms like humans, animals, plants, things like that. So the first step of this process, as you may have gathered, is transcription. Now, transcription takes place in the nucleus, and inside the nucleus is the DNA, which contains all of the genetic information or genes that a cell needs in order to function. This information is stored in a sequence of bases. These bases are adenine, thymine, cytosine, and guanine, or ATCG. You can see them here. A is represented by red color throughout the presentation, T by this dark blue, C by the yellow, and G by the green. And you can see that the information is stored based on a pattern of these as they go up or down the double helix of DNA in an anti-parallel pattern. To unlock this information, though, and begin translation, RNA polymerase comes in, and RNA polymerase unzips the DNA. So you can see here it's in a double helix, and here the DNA, as you can tell here, has been separated into a left strand and a right strand by the RNA polymerase. Now, the RNA polymerase will not only stop there, but it'll travel up the strand of DNA and begin to make mRNA, which is essentially can be thought of as a copy of the DNA, but with one important difference, in that being that instead of using a T, which is this dark blue, it uses a U, which stands for uracil, which will be represented by this purple color. And so RNA will pair up with the DNA, and then it basically becomes, like I said before, a copy of the DNA that the cell can use. And this is important because mRNA, can leave the nucleus, whereas DNA must remain in the nucleus. So mRNA will leave the nucleus through a nuclear pore and enter the cytoplasm, where we will begin the next step of our process, which is translation. So what will happen is the mRNA will come out into the cytoplasm. Here you can see the mRNA is still this orange single-sided strand here. And it'll come to the ribosome, which is this yellow and kind of pink thing represented here. And that's where this ribosome will come into play. And it has two parts. It has a small subunit, which is the bottom, the rRNA, ribosomal RNA, and the large subunit. And those two pieces play different roles in achieving the ultimate goal of the ribosome, which is to provide a place to, well, the ribosome makes proteins, essentially. So the small subunit, what that does is that reads the mRNA, the messenger RNA. So it'll come through, and this small subunit, the ribosomal RNA, will read it in a series of 3. And each 3 is called a codon. And it knows where to start based on the start codon. When it sees this pattern of bases, AUG, that tells the ribosome, okay, we need to start, we're going to be making a protein. And so it'll continue reading in 3s, and each 3 corresponds to something each codon codes for something a little bit, we'll explain a little bit later, and it'll keep reading those until it gets to the stop codon, which is UAG. Now, you can tell that there's something a little bit weird here, and something coming in, the tRNA. And the tRNA has an anticodon, and the anticodon, what that does is it bonds with the codon and provides a place, or a method to bring these amino acids, which are the building blocks of proteins, which is the end goal, remember? And so the tRNA brings the amino acids over to the ribosome, and it knows which one to bring based on the anticodon, and the codon, which tells it to bring a specific amino acid. So this process continues. Obviously, this is a shorter strand of mRNA. Most proteins are very long. This is going to be a little shorter. But it'll go down these codons, and so this first codon will code for an amino acid. This second one will code for an amino acid, this third one, fourth, fifth, but not the stop. The stop just tells it to stop, and so on. It'll go like that. And so what happens is the tRNA comes in, like you can see this one here, and it will drop off essentially its amino acid once it forms a bond with the other amino acids that are already there. And these are polypeptide bonds here. And so that's basically how the protein is made, because remember, the protein is a string of amino acids essentially. And so basically through this process, as you can see here, it's kind of in process. This will we'll end up getting a finished protein. Now these particular sequence of bases and codons will end up giving us this protein here, which has methionine, threonine, alanine, leucine, I believe, and threonine. So yes, that is kind of the process of transcription and translation, which, again, just to reiterate, is the way that a cell is able to take the information stored in the DNA, turn that into something it can use to make proteins in order to carry out its day-to-day life.